Hey guys, today we're going to start talking about 2.4 proteins. So, right now you should have logged into Canvas already. You should have your headphones on. Make sure if you're e learning, you've taken attendance for today. From the homepage, you're going to click on topic 2 and you're going to find 2.4 proteins. So, let's take a minute and take a look, see what that looks like. That's not what we want. We want. Okay, here's your homepage. Topic 2. Give it a second. There we go. So we are on 2.4 now. So here are your understandings, skills, and applications. So this is the list of everything you need to know. So when you're going back through your notes, make sure you have all this stuff. Okay. Now we're not going to do all this today. That's a little too much. So we're going to split it up into 2 class periods. Okay. So we're going to start today and we're going to finish tomorrow or next rotation. Okay. So topic 2.4, proteins. So before we jump in headlong, let's just go back to 2.1, skill 1, because you already had to talk about proteins a little bit and think about them a little bit. So let's go back and think about those molecular diagrams that you had to know. Let's think about the generalized structure of an amino acid. Now remember, amino acids are monomers of proteins, so they are the building blocks. So in each building block of an amino acid, right? So an amino acid is a building block. That building block has 5 parts. There's a central carbon, which is able to make 4 bonds. There's the amino group, which is the NH2 portion of the molecule. There's a hydrogen underneath that carbon because remember, it has to make 4 bonds. There's the carboxyl group, COOH. Sometimes you'll see CHO2. Same thing, it's the carboxyl group. Okay? And last, you have the R group. And the R group is probably the most important part of the amino acid. It's what makes an amino acid different from other amino acids. And that will become more clear in a little bit. So, just to review, those are the 5 parts of the amino acid. Remember, we said it looks like a butterfly? So, here's our butterfly. Okay. So there's 20 different amino acids on Earth, and they collectively build all of the life that you see. So it's kind of amazing to me that there's so much diversity from bacteria to underwater sea life to birds that fly in the sky to us. All of the proteins that build us are composed of pretty much the same 20 building blocks. There's a lot of variation in there and a lot of variety, and we'll talk about how you get that variety using just these 20 different amino acids by the time we finish with this topic. Oh yeah, each of the 20 different amino acids have a different R group. So that's what I meant before when I said the R group is what makes an amino acid different from other amino acids. So there's 20 different R groups, there's 20 different amino acids. So here we have 3 amino acids. This particular amino acid is glycine. That R group is nothing but a hydrogen. This amino acid is called alanine, and that R group is looks kind of like methane, right? But it's not quite. It's a carbon with 3 additional hydrogens. Now you notice the other parts of the amino acid stays the same, right? Next, let's look at our last amino acid. This is valine. And valine's got a pretty big R group. Now you'll notice no matter what kind of R group you have, they're all stable. If you counted up all of the bonds around these carbons, all the carbons would be satisfied. Okay, so let's look at 4 amino acids altogether. We've got serine in the top left, glycine next, then alanine, and then valine. So what's different between them? Well, the amino group, remember this is the amino group, is the same. The carboxyl group is the same. It's the R groups that are different, right? So it's always the R group that differs between different types of amino acids. The butterfly stays the same, that amino group stays the same, that central carbon stays the same, that hydrogen stays the same. It's just the R group that changes. Okay. So understanding number 1 of 2.4. Now we're going to get into the actual lecture part. Understanding number 1, amino acids are linked together by condensation to form polypeptides. So amino acids link together via condensation, that's the same thing as dehydration synthesis, to form a polypeptide. Now you put multiple polypeptides together to make a protein. So you have an amino acid plus an amino acid. They bond together, you get that water that falls out, and you get a polypeptide. Now that peptide links between the carbon of one amino acid, right, and the nitrogen of another because these guys fall out to make water. So this falls out to make water, right? And you have this carbon that bonds to this nitrogen over here. That's really messy. Let's take a look at that again. Okay. So we have an amino acid right here. This is an amino acid, including the R group. And this is an amino acid right here, right? So here's the carboxyl group on this one. Here's the amine group on this one. Here's the carboxyl on this one. So this right here, these together, because it's a condensation reaction, right? So this hydrogen, and this oxygen, they drop out. This is a anabolic reaction because we are building a bigger molecule and it's condensation, right? This water is going to fall out, which means that the bond is going to form between this carbon and this nitrogen. And you can see that right here. And that particular bond between that carbon and that nitrogen, it's called a peptide linkage or a polypeptide bond. And then you get big chains of these together, right? So you would have another amino acid that gets added over here and another amino acid that gets added over here. And those, when you get big chains, they're called polypeptides. Now, these polypeptides, you can think of them as like a chain of paper clips, right? They keep bonding together. Now, they fold and bend into weird shapes based on the R group that sticks out. And that's what provides its three-dimensional shape and function. Now, that's important because in biology, your shape is your job, your form is your function. So, especially when you're talking about molecules, your entire job as a molecule is predicated or dependent on what shape you are. And it's the order of these amino acids that give you your specific shape. So, what you need to do right now is you need to be able to draw a molecular diagram that shows the formation of a peptide bond. Okay, so take a minute. I want you to draw these two amino acids. And remember, you can pause the video whenever you need to. And then we're going to show them linking up. That water comes out. And that is the peptide bond from the carbon to the nitrogen. So when you're drawing peptide bonds, it's important to remember that that water molecule is going to fall out and that bond is going to happen between that nitrogen and that carbon. And if you need to label the parts and pieces, you can, by all means. Here's our carboxyl group. Here's the carboxyl group on this side, right? Here's our R groups, and here's our amine groups. That's kind of messy. But you can see all the different parts. So here's the amino group right here on this side. And here's the carboxyl group. Here's the carboxyl group on this side. And here's the amino group on this side. So you could say the bond between two amino acids is always between the amino group of one and the carboxyl group of another. You never have amino to amino. You never have carboxyl to carboxyl. You always have amino to carboxyl. That water molecule is going to drop out. And then this is your peptide bond. And yes, you will have to draw one of these on your test. So on the test for our first unit, macromolecules, you will have to draw this. So, which of the following correctly shows a bound peptide? We'll see if you can figure it out. Okay. So, understanding number 2. There are 20 different amino acids in polypeptides synthesized on ribosomes. Okay, so ribosomes, think back to like freshman year. Ribosomes are the thing that build proteins, right? Ribosomes are the thing that build proteins. Ribosomes are the thing that build proteins. Let's focus on the first part of this understanding. There are 20 different amino acids in polypeptides, right? That means there's 20 different R groups. 20 different R groups. They can be charged, positive or negative. They can be polar, which would make them hydrophilic and like water, but they don't have to be. They could be nonpolar if they want. They can be uncharged and polar. They can be nonpolar. Put it this way, R groups can be whatever it is they want to be, whether it's charged, uncharged, positive, negative, hydrophilic, hydrophobic. They can be any of those things. I'm not going to make you memorize all 20 R groups. That would be dumb. You should understand that the variety in the R groups is what helps give us the variety of proteins that build the variety of life on this planet. Because R groups are so different, they have the ability to be polar or nonpolar. They have the ability to be positive, negative, or neutral. They have the ability to be hydrophobic or hydrophilic. All those variations allow different types of proteins to be built. And those different types of proteins means they can do different things. And being able to do different things means you can have different types of life. That's the big idea. And it all comes back to these 20 amino acids. Yep, uncharged. Here we go. They can also be special. Cysteine, selenocysteine, glycine, and proline, they're special cases. You don't really need to know why they're special. You just need to know that there are exceptions to every rule, and amino acids are no exception. Here are our special cases. So, most organisms use the same 20 amino acids because, hey, check it out. We all speak the same genetic language. So, every living thing on this planet speaks the same genetic language, meaning a gene from any organism can be inserted into another organism because we all use the same letters A, G, T, and C, and we all use the same 20 amino acids. So, one good example of a widely used protein is collagen. And collagen is the stuff in your skin that makes it springy, right? So, like, you know how when you get a piece of fruit, if you drop it, it gets a dent and it doesn't like spring back? Well, can you imagine if your face was like that? You'd have like dents all over the place, right? Collagen is the thing that makes your skin springy. So if you poke yourself in the face, you don't have a dent in your face. Collagen is also something that you lose as you get older. And that's one of the reasons why you get wrinkles or your skin sags because your body doesn't produce as much collagen to maintain that springy texture in your skin. Now, collagen also provides tensile strength, meaning if you stretch it, it doesn't break. And you find it in your hair and nails and tendons as well. So in your hair, some people have more collagen in their hair than others. If everybody pauses for a minute and you pull like one piece of hair out of your head and pull on it and see how much it stretches. Some people stretch a lot before it breaks, other people's it like breaks immediately. And that's a testament to how much collagen your body puts in your hair, and it's different for everybody. In collagen, there are prolines, and these are types of amino acids that are converted into hydroxyproline, which is not one of the 20 amino acids, right? So this is an exception to this 20 rule because there's exceptions to every rule, right? It's not one of those 20, but it does make collagen more stable. So there are more than 20 amino acids, but they're special cases and they're not used by everybody to build everything. So again, there's always an exception to the rule. Collagen, however, is a protein that you should know. And you can see this is the structure here. And you're probably like, well, how the crap do you get that from like that? So, how do you go from that to that? And remember how I said polypeptide chains are like, they're like paperclip links like this? Well, remember, they all have R groups sticking off of them that make them twist and turn different ways. So, here you can see one polypeptide chain that's twisting and going like that, right? Here we have another one that's twisting and going like that. And that's because of those R groups. So a true protein is multiple polypeptide chains. So a true protein is multiple polypeptide chains. Collagen is a good example of a protein because look at all these different polypeptide chains. There's a whole bunch in there. So, amino acids can be linked in any order, which results in a huge variety of potential proteins, right? So, think if we just take about those 20 with the 20 different R groups that we talked about, right? Think about in math permutations. So, say I have a protein and we're going to make it a small protein. Say it's only 5 amino acids long, right? If I want to figure out how many different combinations of amino acids, I would have to do 20 times 20 times 20 times 20 times 20, right? Let's see what that equals. Where's my where's my yes, I want to keep those. Where's my calculator? Okay. 20 times 20 times 20 times 20 times 20. Holy crap, that's a big number, right? So 3,200,000. I am horrible at this. Here we go. 3,200,000 different amino acids, or I'm sorry, different combinations, right? So it's only 5 amino acids long. Here's the first one, here's the second one, here's the third one, here's the fourth one, here's the fifth one. I could literally have over 3 million different combinations with just 5 amino acids. So let's think about the average length of a protein and different things that live on our planet. So bacterial proteins, they tend to be 320 amino acids long on average. So you'd have to do 20 times 20 times 20 times 20 times 20, 320 times. Eukaryotic, multicellular animals, there's a huge range there, but on average, it's 472 amino acids long. So if this is only 5, right, you can imagine the different number of possibilities that you could get with 427 amino acids. And it's like I said, every protein, its shape is its job, right? So just with 5 amino acids long, here's over 3 million different proteins that could have 3 million different functions. So, when we talk about life, it's very chemically complex, but we have a lot of tools at our disposal to get a lot of cool stuff done. Okay, so next, understanding number 4, the amino acid sequence of of polypeptides are coded for by genes. So, we're jumping around a little bit, but that's okay because this isn't the first time that you've taken biology. You took biology as a freshman. And when you learned about genes, you learned about DNA and you learned about genetic code and you learned about A, G, T, and C, right? So now we're looking at those individual genes. And you probably learned how to turn those genes, how to code those genes through transcription and translation into different amino acids. And that's what this graph is showing you, or this visual diagram is showing you. So the sequence of amino acids in a protein is determined by a section of DNA called a gene. So one gene is one functional unit, one gene, one protein. Genes are the instructions to build proteins. That's what they are. So that's why your genes determine how you're built. They are literally the instruction booklet on how to build all the proteins that are required in your body. So one gene can be a million base pairs long, right? From that information, your body knows how to put together amino acids in the right order. So here we have a gene, and this gene is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 base pairs long, right? Every 3 base pairs, we call that a codon. For every 1 codon, you get 1 amino acid. So if this is 15 base pairs long, right? And it takes 3 amino acids, or I'm sorry, it takes 3 base pairs to make 1 amino acid. That means you're going to have 1, 2, 3, 4, 5 amino acids. Okay, so let's take a look at how this code works. So our first one's right here. GCU. And it says alanine, but how do we get alanine? Well, if you look at this, it's got first letter, second letter, third letter. So our first letter is G, so we look here. Our second letter is C, so second letter, C. So we go this way and we go this way. And now we look at our third letter, which is U, U, come over this way, GCU, and our amino acid is alanine. So that's just a refresher on how we do that, because you should have learned that freshman year. So the genetic code is universal and it's used by all living organisms, and this table is the same no matter what organism you are. And this is how, when we get the genome of a species, we can figure out all the different proteins that are built by that species because we have this table to tell us, using certain genes, this is the sequence of amino acids that you get, amino acids, to create a protein. So I think that's what we just said, right? Yeah. Okay. So I'm going to pause here for just a second. I'll be right back.